One second. The fungi also called this one screen share. See, this is the important characters of the fungi. And another important characters to the fungi is distributed for the characteristic features of the fungi, eukaryotic organisms. These eukaryotic organism means they have true nucleus, which are enclosed, which are to the enclosed the members. These they are non-vascular organisms. They do not have vascular system. And here, the xylem and phloem both are the absent. And fungi have cell wall. The cell wall mostly to made up of to the cell wall chitin only. The fungi cell wall made up of the chitin. Here these are the starting thallophytic plants. That's why the this one observed to the fungi. Mostly all are the thallophyte. The first plant kingdom can be divided into two parts. One is cryptograms and phanerograms. That is the flowering and non-flowering plants. Flowering plants, non-flowering plants can be divided into two parts. One is the thallophytes, can be divided into three parts. One is the thallophytes, bryophytes, and pteridophytes. These thallophyte can be divided into again two groups. One is the algae, another one is the fungi. And fungi. Here, the study of fungi is called, that name is called mycology. The study of fungi is called the mycology. And the fungi eukaryotic organism means have nucleus. They have true nucleus, which are enclosed in the members. True nucleus and here all are the fungi, bacteria, virus. These are the ఏ భవానీ భవానీ ఇది డోర్ తెరిచినావు మూయకుండా పోయినావు నువ్వే తీసినావు ఇందాక these are the total fold into the two types. One is the xylem, another one is the phloem. Your vascular system is totally absent in this one. And with this one, mostly important of the fungi, the characteristic fungi also, this one, the fungi also, characteristic fungi also, the prokaryotic cell. These prokaryotic cell to made cellular and here algae, fungi, all are the made up of different cell wall to make the different chemical structures. In the algae, mostly the cell wall made up of the cellulose like this. But here in the fungi, the cell wall to made up of chitin. Each fungi cell wall, plants also have cell wall, but animals no cell wall. Animal cell is the cell wall is totally absent. And there is embryonic also absent. Embryonic stage also stage of the fungi. And another very important thing, the fungi, this one is the mostly important characters for the and this one that is the most important characteristic features of the fungi. And coming to the next one, they reproduced by means of spores. How to reproduction is there? Reproduced by the means of spores. There are sexual and asexual spores. Sexual spores are oospores, zygospores, ascospores, basidiospores, etc. This is the sexual reproduction. Uh, Next one, depending on the species and condition, Both sexual and asexual spores may be produced. They are typically non-motile. The fungi exhibit the phenomena of alternation of generation. The fungi is the what is the phenomena? Alternation of generation. They have both haploid and diploid stage. Haploid and diploid stage. Uh, these one fungi are achlorophylls. Achlorophyll structure, which means they lack the chlorophyll pigments present in chloroplast in plant cells and which are the necessary photosynthesis. The vegetative body of the fungi may be unicellular or the composite of the microscopic structure. The microscopic thread-like structures are present in the hyphae body. This hyphae can grow from the network that is called the mycelium. Yeast nuclear fungi that do not produce the hyphae. The structure of cell wall is similar to plant, but chemically the fungi cell wall are composed of chitin. Chitin, C18, H13, O5, N. This is the chitin structure. The cell membrane of fungi as a unique sterols and ergosterols. See, sterols are present in the plant sterol, animal sterol, fungal sterol. When the fungal sterol is there, that is name is called ergosterol. Fungal sterol name is called ergosterol. Fungi are heterotrophic organisms. They obtained its food for energy from organic substance, plant and animal matters. The fungi best in acidic environmental, that is the tolerant acidic pH. The fungi digest the food first, then ingest the food. The accomplish this, the fungi produce the exoenzymes like hydrolase, lysase, oxidorine, reductase, transferase. Fungi store their food as such. Biosynthesis of chitin occurs in fungi. Many of the fungi have a small nuclei with receptive DNAs. During mitosis, the nuclear membrane envelope is not dissolved. That way, nutrition in fungi, they are saprophytes. Saprophytic meaning gets energy from dead and recycling manner. That is our parasitism. Some fungi live in a host, attack and kill. Some fungi symbiosis, mutually beneficial cells. Optimum temperature of growth, most saprophytic fungi 20 to 30 degrees centigrade. This is the most important when the temperature is there in the 20 to 30 degree to suitable grow for the fungi grow is that one. And while the 37 degrees for parasitic fungi, all are the parasitic fungi. Growth rate of fungi is slower than that of bacteria. The reproduction in fungi in both sexual and asexual meaning. Both sexual and asexual. Next one, sexual reproduction is referred to as telomorph, fruiting body. Asexual stage is formed to as anarphus, that is called mold-like bodies. The reproduction occurs by both asexual axonomorph, sexual teleomorph mode. Asexual methods, fragmentation, phosomatic budding, fission, asexual spores, formation. Sexual method, gematic, uh, gametic, gametic populations. The gametic and gametangium, that is the population by the gametangium population, stomatic population, and spermatization. This this phenomena, this one is the pheromone is chemical substance produced by the fungi, which leads to the reproduction. Uh, that one is the reproduction, and that is the main important of the fungi. Another character of the fungi, sex reproduction occurs by the both asexual and sexual reproduction. Asexual method, fragmentation method. Fungi consist of a large fragmentations and phosmotic budding, fissions, asexual spores, and formation. Sexual is the gametic population, gametangium populations, and stomatic population, and spermatization. Mostly, fungi constitute a large and diverse group of plant kingdom. The Latin word fungus, Latin word fungus meaning mushrooms. Very highest proteins are there. But fungi are achlorophyllous and heterotrophic thallophytes. These resemble algae in various aspects that have been placed under group Thallophyta. Just like the fungi also same the group. That's why this is called the Thallophyta. The study of fungi is known as mycology, meaning mica. This is the Latin word. The micus meaning mushroom, logos meaning study. Mushroom study is called the mycology, and the scientists concerned with the study of fungi are known as Mycologist. The study of fungi is also known as mycologist. Worldwide, there are about 50,000 to 1,000 species known to be species of fungi. At present, there are about 5,100 genera and 50,000 species of fungi. This one number is constantly increasing because of the ongoing research throughout the world. Various large species of fungi like mushrooms, morels, smut, rods, and rust were known to man since time immemorial. However, their nature, origin, and development were not known at that time. They grow in variety of habitats and show high diversity in their form, structure of the plant, hmm, plant body. Physiological functions and means of reproduction. The plant body is mainly made up of hyphae, which together from the mycelium, except for a few cases where mycelium is either completely absent, mycelium is absent, synchronity, criterium or plant body is unicellular, that is called the Saccharomyces species. The wall is not made up of true cell wall. It is either made up of chitin or fungal cellulose. The fungi cannot synthesize their food from carbon dioxide, water, in the presence of sunlight. The presence of sunlight for decay of organic material. As a parasite, they attack living protoplasm and causing disease of chlorophyll, plants, animals, and human beings. The chief reserve food material is glycogen and oils. The lack of chlorophylls, so starch is not synthesized. Reproduction takes place by means of the function. One second. Hmm. See, this is the fungi. Uh, the introduction of fungi is the that 15,000 to 10,000 species known species. This one is interact from uh, reproduction by this is the absent in the plant body unicellular in the Saccharomyces. The cell wall is not made up of cellulose, two cellulose. In there, made up of chitin or fungal cellulose. Since the fungi cannot synthesize their food from carbon dioxide, water, in the presence of sunlight. Their mode of nutrition is saprophytic, parasitic, and symbiotic. As a saprobe, they are responsible by decay of organic material as a parasitic and symbiotic. As a spoke, they are responsible for decay of organic material. As a parasite, they can, as a parasite, they can uh, parasite and they attacked living protoplasm and causing the disease of plants, plants, animals and human beings. The chief reserve food material is glycogen. Chief reserve food material is glycogen. These are the lack of the reserve food material. They lack chlorophyll So starch is not synthesized. Reproduction takes place by means of vegetative, asexual, and sexual reproduction. What are the occurrence? Occurrence, fungi are cosmopolitan and most diversified in nature. They are found in almost all habitats where a life is possible. Some fungi are terrestrial, some are airborne, while some occur in fresh as well as marine water. The members of Phycomycetes are found in water and are known as aquatic fungi. The fungi to develop in the underwater, that's where that type of fungi is called aquatic fungi. Fungal species are also found in epiphytic state on algae and some other aquatic plants. Some of the fungal species grow on dead organic material present in water. Some species are found under the surface of each and are subterrestrial in nature. A few of fungi are also found as endosaphytic fungi in leaves and the stems are also healthy plants. These fungi can be categorized between beneficial and harmful species. Some produce secondary metabolites like proteins, protect the host from the insects, grazing animals, while other can be toxic and reduce the crop yield. The fungi are microscopic as well as macroscopic. Macroscopic fungi such as mushrooms, puffball, bracket fungi, cup fungi, and morels are recognizable with naked eye even from a distance. However, the microscopic fungi can only be identified with the help of cup microscope by the subject experts. Okay, these are the identification characters from the fungi. Various species fungi are parasitic, also infect a variety of plants, animals, and human beings. The parasitic fungi, which grow on vascular plants, like plants causing various diseases in their fur, example, brown patch, canker, downy midule, clubroot, damping of anthracones, bottom rot, crown rot, etc. And, and are responsible for economic loss. Ultimately, al- elementary canal of human beings and mammals are also occupied with fungal species where they causing stomach disorders. Some of the fungi species cause skin diseases such as athlete's foot, mycosis, ringworm, onychomycosis, candidiasis, etc. In this way, fungus were are diversified and accustomed to usual habitat. Characteristic of fungi different from other kingdoms. Other characteristic of fungi different from other kingdoms. There are a number of characteristics that make fungal kingdom different part from plant, animal, and other kingdoms. Some of them are follows. The first one, fungi body is unicellular and is more often composed of microscopic threads that is called hyphae. Thread-like structures too found into the fungi. That name is called the hyphae. That name is called hyphae. They are non-vascular bundles due to the absence of any specialized transport system, specialized any transport tissues. They are heterothalic and feed on performed organic material. Like animals, their stored food material is glycogen, whereas plant stored food material that is called the starch. And unlike animals, fungi first digest for the food with the help of coenzymes and then insides, etc. Next point, majority of fungi responses by means of spores that are being dispersed depending upon the environmental condition, both asexual mitotic division, sexual is the meiotic division. Meiotic meaning meiosis. Spores are produced. In this life cycle of fungi, only a few stages are motile, such as zoospores. Structurally, cell wall of fungi is similar to the plant, but it is chemical composition, the indifferent. Fungal cell wall made up of chitin. This chitin that beta 1,4 linkage in acetoglycose, one linkage of N-acetylglucosamase in contact to plant cell wall, which are composed of the beta 1,4 linkage of glucose. Fungal cell member have unique sterols. The sterol name is called ergosterol. In contrast to any animal which have cholesterol. Ultrastructure of cytoplasm is quite similar to plant cells, but there are differences in which and structural of organelles. Nuclei of finus is very small and they have only few chromosomes. Last while meiosis and mitosis occurs inside the nuclear envelope. Coming to the thallus. Range of thallus. Fungi is the unicellular organism. First one is the yeast. The fungi are eukaryotic organisms, which are unicellular, can be multicellular or acellular also. Yeast is of the wide occurrence, is found on sugar surface of refined fruits and can be grow easily grow in any sugar solution. Individual cells remain attached to either to each other, forming a chain. Fine structure of each cell resemble with that of a eukaryotic cell. The cell has a well-defined nucleus, endoplasmic reticulum, mitochondria, and other organelles, along with a large area of the cell occupied with the vacuole. The cell wall is made up of 2 to 3 layers thickness, compressing of glucans and mannose. Variable amount of proteins and lipids are accumulated in the cell depending upon the stage of development. They have a particular ability of utilizing carbohydrates and its same Saccharomyces is applied to this group. This is the uh, Saccharomyces cerevisiensis. And slime mold. During a certain stage of the life cycle of slime mold is a unicellular. Multinucleate forms are also seen. However, these are not considered true fungi since their characters resembles to both protozoa and fungi. It becomes very difficult to characterize them easily during the course of their life cycle. These organisms shows protozoan like unicellular and multinucleate or fungus like stage. These multicellular slime molds are two types. Cellular type, plasmoidal type. During the vegetative phase, Dictyostelium, Discoideum, a small independent uninucleate cell known as Myxomobia. It feeds the bacteria by ఇది కూడా charging పెడితే పెట్టు. The cell type is the Dictyostelium discoideum. Rima, Rima. Uh, a small independent uninucleate cell known as myxomeba. It feeds on bacteria, phagocytosis, and cell multiplication occurs by binary fission. During the last stage of development, a large number of myxomoeba comes together to form a single multinucleate slug, but the individual cells remain their cell membrane intact. The resulting structure is known as pseudoplasmodium. Like a fungi, during reproduction phase, spores are produced in a sporangia. Here, all reproduction time, the spores to produced sporangia. Each spore upon germination gives rise to an amoeba-like structure. Plasmodial type during vegetative phase in uh, Echinostelium minutum. There is a formation of large mass of multinucleate amoeboid cytoplasm where diploid nucleus, the individual cell is the individual cells are, however, not delimited by a cell membrane. It feeds on bacteria and encycled myxomoeba. They do not have a defined size, defined structure, and defined shape. That's why they may be globous or spreading like a sheet over a large area in form of a thin network. It alters its shape depending upon the substratum and engulfs food on its way. During reproduction phases, entire plasmodium take part in the formation of fructification which bears spores. The spores germinate to form flagellated cells, which later on develop into plasmodium. The slime mold, plasmodium is brilliantly colored. Plasmodium, such as Physarum phylocephalum, is structural less as granule, vacuole, and other bodies embedded in it. Additionally, one of the most facing process about facing forward about uh, 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 facing problem processor under electron microscope. Slime mold is the class such as Physarum phylocephalum is structureless as granules, vacuoles, and other bodies embedded in it. Additionally, one of the most facing process about plasmodium is the protoplasm streaming under microscope. This streaming is related to the presence of contractical protein known as myxomyosin filamentous forms. Most of the fungi are filamentous, and each filament as known as hyphae. That is called the basic unit of fungal body name is called hyphae. Basic unit of fungal body name is called hyphae. Mass of interwoven hyphae is known as mycelium interwoven, which has a large surface to value ratio close to food source adaptation for absorptive mode of nutrition. See, this diagram represent of the life cycle, acellular slime mold, this one, dictyostelium, discodium, amoeba-like stages. These are all amoeba-like stages. Here, aggregation of amoeba, cellular plasmodium, aggregation of amoeboid, migration stage or slug stage. This is the fruiting body. These are the fruiting bodies, spores and germination, plasmodial slime mold, echinocystelium minutum, young multinucleate plasmodium, mature plasmodium, fructification, formation of sporangium on fruiting body meiosis, germinating spore, myxomebia or gamete formation, fusion of gametes and plasmogamy in myxomebia. Next one, pseudoparenchymatous form. Here, as it has been already mentioned above that fungi, mycelium forms a network of loosely interwoven hyphae. In some species, specific group of fungi, the entire mycelium or some part of the fungal mycelium undergoes a various modifications. The hyphal mass in cross section appear to be continuous structure as the wall of hyphae fuse and they lose their individuality. This structure looks like the parenchyma tissue of higher plants, but it is not true parenchyma. Such tissue is known as plectenchyma. Details are in, that is called the plectenchyma. Next, how to organize the thallus or vegetative structure. Fungi shows a great diversity in the thallus organization. Their thallus range from single-celled structure to massive multinucleate structure. Except the unicellular forms, most of the fungi are made up of thread-like filamentous. The structure name is called hyphae. These hyphae branch profusely and form a network-like structure is called mycelium. These mycelium unicellular thalli often gives rise to pseudomycelium, which is formed when cells produce buds in succession fail to separate from the parent cell and remain attached to either other in a easily detachable chain. However, in some fungi, pseudomycelium is formed in response to high sugar concentration. Such fungi is mucorales. Some fungi may show dimorphism in their thallus organization. as they may either exist in unicellular or filamentous form. The change in the thallus organization from filamentous to unicellular form is brought about by the alternation in environmental condition. Environmental condition. Some of the fungi, some of the zoopathogenic fungi are mycelial in culture but become unicellular and yeast-like in the host. On the other hand, certain phytopathogenic fungi that is a phytopathogenic meaning smut diseases, leaf curl fungi become yeast-like thallus culture or the mycelial host. Hmm. The fungal thallus grows centrifugally as the younger and more active part are present towards the periphery and older thallus is found towards the center. At the end, all thalli from reproductive structures. In unicellular fungi, whole thallus may get converted to reproductive structure. Such thalli are known as holocarpic. However, in most of fungi thallus is different into vegetative and reproductive part. This type of thallus is known as eucarpic thallus. Coming to the hyphae. Fungal hyphae is microscopic structure which are bound by a ridge cell wall, contains protoplasm and may be hyaline, uncolored or dark with brown and yellow type pigments are present. It grows indeterminately only at the tip and has a fairly constant diameter of approximately 2 to 20 millimicrons. The thallus of higher fungi has the capability to unstones during this process. Neighboring hyphae in the thallus put one short branches which make contact the tip and with the time may establish contact with each other. With the time, well of the hyphae get dissolved at the point of contact and form a continuous tube five joining the two. This process includes efficient cytoplasmic growth, exchange of nuclei, create a strongly mycelium network, and coordinated growth of tissue. The branching is monopodial as the apex of the leading hyphae is not suppressed and it grows along with the most active leading branch. Lateral branches are usually singly formed but may be formed in pair or opposite also. Sometimes these lateral branches may arise in whole of the three or more are known as verticillate. In the members of Zygomycetes or Oomycetes, hyphae are long, multinucleate, and comprises of continuous cells called cenocytes. These hyphae are non-septa or aseptate. Even in these cases, the septa are formed to delimit aged hyphae, or when there is a formation of reproductive structure. In Ascomycetes, Dideromycetes, and Basidiomycetes, or the higher fungi, the hyphal protoplasm is interrupted at regular intervals by crosswalls or septa. That is the septum hyphae in the case is known as septata hyphae, but the protoplasm is still continuous throughout the fungal hyphae because of the perforation which are present in the cross walls. Diagrammatic representation fungi. Septa are two types. One is the primary and second one adventitious. All types of septa are formed by centripetal growth from the hypal wall towards inward. Primary septa is formed as result of nuclear division between daughter nuclei. However, the formation of adventitious septa is independent of nuclear division and is formed because of the changes in cytoplasm concentration as it is moves from one part of the hyphae to another. In ascomycetes, cytoplasmic continuity is maintained by a small simple pore in the center. This is the septa, where perforated septa are also found in Basidiomycotina or Basidiomycetes, except rust and must. However, which slightly modifications which involves the presence of a barrel-shaped inflammation with a hemispherical performed membrane on each side of the opening. This is known as dilomorphism or dilophore septum. Fungal mycelium is called homokaryotic when individual cells of the septae have hyphae have genetically identically nuclei. On the other hand, some fungal mycelium contains nuclei of different genotype which arise as a result of mutations or anastomosis of hyphae. These hyphae are known as heterokaryotic type. The members of Basidiomycetes may have two genetically different nuclei. One is the dikaryotic, may have single haploid, genetically identical nuclei in each segment. That is the monokaryotic. This is the dolichos, aggregation of fungal hyphae. At a certain stage in life history of all fungi, they show various degrees of hyphal aggregation, ranging from loosely to compactly woven tissue. All such organized fungi tissues are known as parenchyma. The three general types of plectenchyma, prosenchyma, pseudoparenchyma, pseudosclerenchyma. Prosenchyma, it is loosely interwoven tissue in a which component hyphae lie more or less parallel to one another and their elongated cells are generally distinguishable from each other. Second one, pseudoparenchyma. It consists of closely packed isodiametric or oval in shape. This one thick ring This is the thin-walled cells. Has a close resemblance with parenchyma of vascular plants. Pseudosclerenchyma, it consists of closely packed and thick-walled dark cells are present. Plectonchyma form various types of vegetative and reproductive structures such as mycelial strands, mycorrhizal, rhizomorpha, stroma, sclerotia, and sporophore. What is the mycelial strand? They are found in Basidiomycotina, some members Deuteromycotina. These are the aggregates of parallel and comparatively undifferentiated hypha. Mycelial strand is formed around one or more leader hyphae. These leader hyphae grow out of the thallus margin, become surrounded by their own interweaving and anastomizing branches to form a crowd, 1 to millimicron thick and few centimeter long. The mycelium strands are involved in translocation of materials, and these are the means or by which fungus is capable of extending itself to a new substratum from an established food base. The mycorrhiza is the symbiotic associates between fungal hyphae and plant roots. The root tip of coniferous and deciduous plants are often made up of several layers of fungal cell, fungal mycelium, extend into the layer of soil and then inverts in the cortical cells of the root to form Hartig network. The extended fungal mycelium replace the functions of the roots and is thus involved in better absorption of mineral forms the soil. The roots which mycorrhizal network outperform root without mycorrhizal associations. Rhizomorpha meaning the shape of the root, the root shape, are highly differentiated root-like hyphal aggregation which have a well-developed apical meristems, central core, thick-walled elongated cells, and rind, smaller thick-walled, highly pigmented cells. These are produced by Armillaria mella, a tree and shrubs parasite. Rhizomorphous helps the fungus in spreading from one root system to another root system. The stroma is a compact hyphae aggregation like a cuisine or matters in which fructifications are formed. These are reported in members of Deuteromycetes, Basidiomycetes, and Ascomycetes in the form of various types of ascospores, pycnospores, basidiocarps, arvelli, cinnamata, pycnidia, and sporodokia. These are the stroma. Next one, sclerotium or sclerotia. Sclerotia is a hard resisting body formed by aggregation of stomatic hyphae. They may be round, enclosed, or inflated. Mass are the characteristic feature of a particular species in size, in shape, in structure, and color. Microsclerentia. Microsclerentia meaning macrophomenia facioli. Macrophomenia facioli. So, maybe less than 9, 10, and less than 100 millions meters diameter, and majority of sclerotia do not exceed behind a diameter of 2 centimeters. In Porphyria mycelia, selection are more than 25 centimeter in diameter and weigh around several kilograms. These are around the several kilograms. These are these are meant for storage and help the fungus in survival during unfavorable environmental conditions by acting as a propagulate. Next, coming to the last one, sporophores. Sporophores are the spore-bearing structure, which are usually erect and aerial, erect, aerial, branched. When the branching are there, Pyronospora or unbranched albugo and beer sporangia albugo or conidia peronospora on them. The sporospora being sporangia are called sporangiospores, and thus they bear called known as corneliopores. Sporophores may often present in groups and from Pycnia, Hymenea, Sporodokia, and Acervally. This is a sporo. Coming to the ultrastructure of fungi. The unicellular yeast as well as filamentous fungi as a typical eukaryotic cell organism. As other non-photosynthetic organism, fungi contain all majority organelles except the chloroplast, mitochondria, ribosomes, endoplasmic reticulum, nucleus, nuclear membrane, Golgi complex, ribosomes, nucleus, only nuclear membrane is absent. All character, microbial character, all are the microbial character. These unicellular yeast as well as filamentous fungi as a typical eukaryotic cell organization. As other non-photosynthetic organism, fungi contain all major organelle except chloroplast. There are a wide range of unusual membrane-bound organelle, site of the special function, and other organelle which are the site of the special functional fulfill specific requirement or helps in adapting suitably to a constant growth habitat. The cells of vegetative hyphae are thin-walled, 0.2 millimicrons, and rigid. It serves as a number of important functions such as provide mechanical, first one, to provide the mechanical strength. Provide the mechanical strength and helps to fungi in assuming a variety of forms such as a penetrative, ramifying, and proliferating cells. Second one act as a molecular sieve, uh, regulates the movement of the large molecules. Determine the characteristic shape, size of fungal spore. Provide protection of the cell. Osmotic lysis, wall is permeable. Okay, sir. I'll end the class.